everyone, my name is Sharon Platt. I am one of the science tutors for the Student Success Commons, and today I would like to go over protein synthesis. It's a fairly complicated process, and I'm hoping to make it as easy as possible. So I have a bunch of little random household objects that I'm going to use to see if I can make it better. Alright, so protein synthesis, we are going to start with DNA, and as I'm sure you remember, DNA is housed in the nucleus of the cell. And I do apologize, I do not have the book you guys have. I'm just using the books I have, so hopefully they're similar enough you get the idea. So DNA is in the nucleus, and that is the pattern that is used for everything that we need in our body. And then we're also going to talk a little bit about the rough ER right here. And it's called rough because I don't know if you can see, there's little tiny orange dots on there. Those are called ribosomes. And we're going to talk quite a bit about ribosomes, a little bit about the rough ER, and about the nucleus. All right? So, here is my very crude model of DNA. We know that it has two strands and it's a double helix, and I tried. Please look in your book and look at the pictures. They're a whole lot better than my models. But we'll let this represent DNA for now. And there are the two strands, and when something gets copied from the DNA, because we don't want the DNA to leave the nucleus, that would cause all kinds of problems that I won't go into here, but the DNA needs to stay in the nucleus. So when we need to make something from it, we need to make a copy. So we're going to take a section of the DNA, and there's going to be an enzyme that's going to open out the DNA so that other nucleotides can get to it. You remember that DNA is made up of 4 nucleotides, cytosine, guanine, tyrosine, and adenine. And then mRNA has uracil. All right. So we have those 4, and they go all along here. And then more are going to come in when this is opened and create mRNA. And we call it mRNA because it's messenger RNA. So this is going to be our copy. So it comes in and it forms, first there's nothing. And then it forms as the copy goes along here, and then it comes out. And it gets chopped up a little bit, some pieces taken out, and the other pieces spliced together, and it makes a slightly shorter chain. We're not going to actually do that part of it, but you do need to know that that happens. And I don't understand all the things that control why that does that. But I read a little bit about it today. It's fascinating, but it's above my ability to explain. All right, so that's our mRNA. So let's take a closer look at DNA for a moment. And we're just going to go with the letters. We're not going to do cytosine, guanine, all of those. We're just going to go with the letters. All right, so this is our DNA, double-stranded. We've got our template strand down here and our, what we call a coding strand up here. That's what we call the other coding strand. The other thing is sometimes one of them is the template strand and sometimes the other is. It depends on the gene. But whatever template strand a gene uses, it always uses that strand. So this particular gene is always going to use the red strand. Some other gene might use the green strand. All right, so that's what those are. Okay, so we have our letters here. And so when we make our copy, we're going to take from the template strand, and others are going to come match it. The complementary nucleotides are going to come match this. So we're going to get a G, and then an A, and then a C, and then a U, not a T, because all the T's change to U for our mRNA. All right, and we're just going to keep going right down the line, and it's going to make a nice little copy for us. Okay, the other thing we need to know is in the DNA, every 3 is called a triplet, every 3 letters. That's how our language works. All the words have 3 letters in DNA. And this is why it's so important that we start in the right places. So this first codon, and I did not do this correctly, this is just a made-up thing. This first triplet is going to make a codon that is a certain code that's always the same one. It's a start codon. So that tells everything where to start when we're creating it. All right, so this is our copy. This is our mRNA, and this is our copy. And you see we have a G and an A and a C and then a U all the way down, just the complementary ones from our template strand. And these on the mRNA, the triplets are called codons. On the DNA, they're called triplets. On here, they're called codons. It's just a different name because the letters are slightly different. We can't call it the exact same thing and differentiate which we're talking about. All right, so this is a codon, these three-letter words. All right, and so this, going from DNA to RNA, is called transcription. Think script, you're writing, okay? You're just rewriting with minor changes from DNA to mRNA. Just making a copy, that's all you're doing, okay? Same language. We're still using nucleotides, all right? Then with the mRNA, these codons, they actually code for a specific amino acid. And let me see if I can show you this. I don't know if I can get it close enough that you can see it very well. But this is the chart that we use. And you see there's your start codon, AUG, and there's all these other codes. This chart can be a little confusing. Basically, the first letter in the code comes from this side. So all the guys here start with U. All right, all the guys here start with, what is that, C? I can't tell. Yeah, the C, okay? All the ones in this line start with C. The second letter comes from up top here. So this one is going to be UU and then something else, all right? And then this one is going to be UC and then something else. And that third one comes from over here. So we're used to charts that have 2 sides, and you know, kind of like a Punnett square, and you fill in with 2 things. This side chart has 3, so you have to take first letter from here, second letter from up here, third letter from there, and that makes your 3-letter code or your word. All right, little confusing, but hopefully that will help. Okay, so now we have our mRNA. We're going to let this little guy represent all these little letters, all right? And the mRNA, after it's all been spliced and everything, comes out of the nucleus, out into the cytoplasm, and it finds a ribosome. A ribosome is made up of 2 pieces, a large subunit, a small subunit. And when they meet mRNA, or the mRNA meets it, I'm not sure who says hi first, they come together, and the 2 subunits, the mRNA goes in between them, all right? And the first thing that's on that mRNA is that start codon. And so that start codon comes into this first little section here, this first little space, and there is going to be a match for that, obviously, all right? So we're going to use these little clothespins to be the tRNA, or the transfer RNA. So we've got 3 RNAs. We have messenger RNA, transfer RNA, and ribosomal RNA. Okay, little confusing. They're all made up of similar things, but they function differently. All right, so this transfer RNA, it has an amino acid here, and it has an anticodon over here that matches, or it complements. It's the complementary codon. It's the anticodon to the codon. All right, so that start codon comes in here. If I can grab it. With our mRNA, start codon comes in here, and then the tRNA says, oh, I recognize that. I complement that. Okay, I'm going to go down, and I'm going to be right here. And then our mRNA moves over a block, and the next codon is then under this one, and another tRNA will come and sit there. But we're going to follow this guy for a minute. Eventually all three will be full at once, but I don't have enough fingers to do that. So in this middle one here, this is where the peptide bond is formed. So this start codon is the first one, and then others will come after it, obviously, and we're going to start to build our polypeptide, okay? And as it builds, it's going to come on up out here when there's no room for it in there anymore, okay? So now our little tRNA, he's let go of his amino acid, so he's going to move from here after it's gone over to here, and then he's going to exit. All right, so you always have one coming in here, one here forming the peptide bond, and then moving on over to here, and that one exits. Come in, peptide bond, exit. All right, that's what those three are. And they keep going all together, all right? So I can't hold them all together or I'd show you. All right, so as this keeps going, we keep getting tRNAs, and they keep bringing more amino acids. And then the ribosome puts a peptide bond in there, binds those two together. Then we get another one right here. And I only got 6 of these. And usually there would probably be hundreds of these amino acids in a chain. But I only did 6 because I thought that was a little easier to handle for a demonstration. All right. So now we've got our polypeptide chain. All right. And it comes up out of the ribosome as it's done. And when it's done, there are stop codons that'll come into here, and then it'll know it's time to break it off and let it go. And so it goes out, and sometimes it goes out into the cell. It just comes out and it does its folding. Maybe you remember that from earlier in the course. Proteins fold various ways and form little bonds in between the amino acids there. And they form shapes that make them usable, all right? And then sometimes it comes out of the ribosome and it goes out into the cell and it does its job. Other times, while it's still being created, the ribosome will go over to that rough ER and attach there, and it'll feed it into the rough ER, and the folding finishes there. And it just depends on the purpose of the protein, where it's going to go and what it's going to do after. Fascinating. I just read up on that today, but I won't go into it and confuse you too much. All right, so our protein gets folded, and then it is ready to go, and it goes out into the cell and does its job.